dPhagemid vectors. 1. Phagemids. Sometimes generation of a single-stranded DNA becomes important for DNA sequencing and site-directed mutagenesis. Using a single-stranded DNA containing Bacteriophage M13, a series of cloning vectors were developed. The bacteriophage fluid is closely related to M13, which also infects E. coli. Phagemids are plasmids that contain origin of replication, ORI, for single-strand DNA containing bacteriophage such as FL. E. coli maintains a plasmid as double-stranded DNA due to plasmid ORI gene. If E. coli cells are infected by the helper fluid phage, the ORI-OFFL is activated. It switches to a mode of replication and produces single-stranded DNA which is packed into phage particles as they are extruded from the host cell. Figure 5.14 shows a Phagemid pBlue Script 11 kS, plus or minus, which is used for generation of single-stranded DNA molecules. A. pBlue Script 11 kS, plus or minus. This vector is 2961 BP long derivative from PUC19. The Ks denotes the orientation of polylinker, i.e. transcription of latch genes proceeds from the KPNL restriction site towards SACL restriction site. It consists of multiple cloning site MCS, flanked by T3-T7 promoters in opposite directions on the two strands, figure 15.14. The MCS is set up for latch blue-white screening. A LACL promoter complements with latch of E. coli upstream of latch region. This feature helps in selection of recombinant vector based on the criteria of development of blue-white colonies. White colonies are formed if foreign DNA is inserted in the vector. It consists of FL+, and fluid origin of replication obtained from a filamentous phage FL. It is used to get sense, plus, and antisense, strands of latch gene after co-infecting the host with a helper phage. But in the absence of the helper phage and origin of replication, colal ORI from a plasmid is used. Moreover, for antibiotic selection of recombinant Phagemid vector, AMPR, ampicillin resistance gene, is also used. A number of restriction sites are present in these genes. See the figure. Figure 5.14. The Phagemid pBlue Script 11 kS plus vector. Yeast plasmid vectors. Yeast Saccharomyces cerevisiae is a unicellular eukaryotic microorganism has its long historical importance. It has been used for production of alcohol and alcoholic beverages. It can easily be cultured on artificial media. However, its genetic material is well organized into chromosomes present inside the nucleus. Yeast cells contain their own plasmid known as 2 micromole circle plasmid, which is used as vector for foreign genes. It is present in many stains in 50 to 100 copies per cell. The foreign DNA taken up by yeast is integrated by a specific crossing over to yeast DNA containing a homologous sequence on chromosome recognized by the yeast as the origin of replication ORI. In some cases, segments of yeast DNA linked with foreign DNA have been demonstrated to transform very efficiently, about 104 cells. The transforming molecules replicate autonomously within the cell. DNA sequences of such property are known as ARS, autonomously replicating sequence fragment. ARS fragments have also been isolated from a number of eukaryotic organisms such as Coenhaurabditis elegans, Ditiostelium discoideum, Drosophila melanogaster, Z. maize, etc. However, plasmids have been constructed by using ARS fragments when joined at centromeric sequences, CEN region of about 6 to 10 kb DNA, to ensure the replication and stability in yeast. CEN is needed for DNA molecule to segregate correctly during meiosis and mitosis. Sometimes the minichromosomes are lost during meiosis when they are attached by chance to spindle apparatus. Thus, CEN regions of yeast plasmids are useful fragments because they confer stability. In 1977, Ratskin and Carbon inserted a segment of yeast DNA into Col1 plasmid. They found some of the E. coli strain lube as leucine-independent transformants. This showed that the segment of yeast chromosome consisted of the gene for yeast enzyme which was absent in E. coli strain lube. After ligation, the mutant E. coli became able to grow on normal medium. Ahinen and co-workers in 1978 found that Col1 plasmid containing LEU2 gene, wild type, can transform LEU2 mutant yeast's pheoplast to form leucine-independent phenotype. It is customary with yeast to write wild type allele in capital letter, e.g. LEU2, and mutant allele in small letter, e.g. LEU2. The LEU2 gene was added into the yeast chromosome and transferments were produced. In this way, several yeast genes were isolated and integrated to produce transferments. Such yeast genes are LEU2, URA3, TRPI, and ARG4. Thereafter, unique markers were inserted in wild-type genes, while introducing the foreign DNA into yeast cells for further selection. The eukaryotic genes consist of introns, hence, these genes rarely function in bacterial cells. In contrast, being eukaryotic microbe, most of yeast genes containing promers successfully function in bacterial cell. The difficulty associated with yeast was the transformation at low frequency due to crossing over integrative transformation. Therefore, several plasmids were developed to solve the difficulty, such as YLP, YEP, YRP, YCP, YLP, pYAC, shuttle vectors, etc. Figure 5.15. 1. Yeast integrative plasmid YLP. The YIP consists of only a yeast marker added to bacterial plasmid. It lacks origin of replication. Hence, it cannot replicate inside the yeast cell without integrating into a chromosome. Therefore, its integration is must for successful transformation. It results in low-frequency transformation of yeast cells yielding a transferment per gg of DNA molecule. Integration takes place due to homologous recombination between the inactive gene present on one chromosome and yeast gene carried by YIP as the selectable markers. Crossing over takes place between the chromosomal LEU region and LEU2, carrying plasmid. Consequently, the flanking region linked to plasmid is integrated to LEU2 region. Autonomously replicating system, ARS, is absent in that plasmid. Therefore, YIP cannot replicate as a Phagemid, figure 5.15a. 2. Yeast episomal plasmid, YEP. The YEP is the two-circle-based vector which replicates autonomously without integrating into a yeast chromosome. The 2-pun plasmid is 6 kilobits in size with so high frequency for transformation. The 2-GM DNA consists of origin of replication and representative genes. This plasmid results in high-frequency transformation, i.e. about 2x104 transferments per ggDNA. The number of restriction sites is limited. It is found in high copy number in the cells. It will lose its stability if 2 sequence is lost. It is moderately stable in the cell, figure 5.15b. See the figure. Figure 5.15. Different types of yeast plasmid cloning vectors. 3. Yeast replicating plasmid YRP. On the basis of chromosomal elements, this vector has been developed to support autonym 011s replication of plasmid in yeast seals. ARS was isolated from many regions of genomes of yeast and other eukaryotes, and ARS-based vectors were constructed. ARS-based vectors are very unstable but can form high copy number inside the cells, figure 5.15c. 4. Yeast centromeric plasmid YCP, or yeast centromere CAN. YCP consists of a sequence of chromosomal centromeric DNA of yeast. It acts as true mini-chromosome which becomes circularized. It increases the stability of plasmid and segregates during mitosis and meiosis. Copy number of this plasmid is useful when low copy number of cloned genes are required, figure 5.15d. 5. Yeast linear plasmid YLP. It is a linear plasmid constructed by adding telomeric sequences to both ends of a cleaved ARS-based plasmid. Telomere is the end of chromosome. Telomere was isolated from a ciliated protozoan, Tetrahymena. This linear plasmid is present in single copy as a chromosome and are very stable and cannot replicate in E. coli. Therefore, it has drawn less attention but has raised much possibility to put together the complete set of genes in artificial chromosome of eukaryotes, figure 5.15e. F. Yeast artificial chromosomes YAC. The plasmids can accommodate about IOKB foreign DNA, phage can accommodate about 20 kilobits DNA insert, Cosmid can accommodate about 40 kilobits long 4 DNA segments. But for stable replication and segregation in eukaryotes, particularly in yeast, a small and well-defined sequence should be constructed as recombinant chromosome. With this objective, David and co-workers developed, in 1987, yeast artificial chromosomes YAC, by using new approaches where DNA segment of several thousand base pairs, about IMB, can be cloned. Initially it was used for investigation of the maintenance of chromosomes in the cell. Later on it was used as vectors for carrying very large cloned fragments of DNA. It has also been used for physical mapping of human chromosome in Human Genome Project. YAC consists of centromere element CAN, for chromosomal segregation during cell division, telomere, and origin of replication, ORI, were isolated and joined on plasmids constructed in E. coli. Structure of typical YAC3 vector is shown in Figure 5.16. See the figure. Figure 5.16. Yeast artificial chromosome YAC3 cloning vector. The YAC3 vector contains the E. coli origin of replication ORI, and a selectable marker AMPR, a yeast DNA sequence, genes each for uracil biosynthesis pathway URA3, providing centromere function CAN, autonomously replication sequence ARS, which acts as yeast ORI synthesis pathway TRP, and telomeric T sequence, which is extended by the enzyme telomerase inside the yeast cell. Figure 5.16. The TRPI and URA3 are yeast selectable markers, one for each end, which ensure that properly reconstituted YACs survive in yeast cells. SUP4 is inactivated in recombinants and acts as a basis for red-white selection, unlike blue-white selection in E. coli. There are recognition sites for restriction enzymes such as SMAL and BAMHI. Before cloning, YAC is digested with restriction enzyme, BAMHI and ECRI, and recombinants are produced by inserting a large fragment of genomic DNA. This molecule can be maintained in yeast as YAC. The transformants that contain YAC can be identified by red, white color selection. Non-transformed yeast contain white colonies. Red colonies of yeast contain recombinant YAC molecules. Due to insertion of DNA molecule into EcoR1 site, SUP4 gene is inactivated and no protein will expressed. This facilitates to develop red colonies. Bacterial artificial chromosome BAC. The BAC was developed by Mel Simon and co-workers as a cloning vector system in E. coli and an alternative to YAC vector. BACs are maintained in E. coli as a large single copy. These are used as vectors for cloning very large, greater than 50 kilobits, sequences of DNA. Sometimes these fragments turn out to comprise non-contiguous, non-adjacent, segments of the genome and frequently lose parts of the DNA during propagation. Hence, these become unstable. BAC are constructed by using fertility or F factors resent on F plasmid, of E. coli. BAC vectors contain ORI gene, REAP gene for maintenance of F factor, genes para, B, C, for plasmid copy number, an antibiotic resistance gene e.g. chloramphenicol acetyltransferase CHLR gene for selection of plasmid and many restriction sites for insertion of foreign DNA. The F factor encodes its own DNA polymerase and is maintained in the cell as one or two copies. BACs are maintained as single copy plasmids in E. coli, excluding the replication of more than one BAC in the same host cell. The upper limit of foreign DNA to inserted in BAC is about 300 to 3,500 kilobits. BACs are alternative to YACs and, therefore, used in genome sequencing projects. See the figure. Figure 5.17. Diagram of below BAC 11 vector and cloning and selection of genomic DNA by using this vector. The method of preparation of BAC library is the same as for plasmid library. But here the DNA insert is prepared by pulse field gel electrophoresis PFGE. PFGE has made it to separate, map and analyze very large DNA fragments. Large-sized globular DNA migrates easily after applying discontinuous electric field. Example of a BAC vector is below BAC 11, figure 5.17, which is partially digested while cloning a DNA fragment. Genomic DNA is partially digested with Hind3. Size fractionation of DNA is made by PFGE. Similarly, below BAC 11 is treated with Hind3 and phosphatase. Such digested vector and fragments of genomic DNA, 100 to 300 kb, are ligated. The recombinant plasmid containing DNA insert is transferred into E. coli cells through electroporation, see Chapter 6. E. coli can be transformed very efficiently by BAC. Then E. coli cells are plated on pound medium containing chloramphenicol, IPTG and X-gal for selection. White colonies of E. coli are picked up based on chloramphenicol resistance. In 1989, by using this vector, DNA segments from bithorax gene of Drosophila have already been cloned. BAC clones are stable for many generations. In contrast, it lacks positive selection for clones that contain DNA inserts. Therefore, the yield of DNA is very low. Plant and animal viruses as cloning vectors. Many eukaryotic genes and their control sequences have been isolated and analyzed by using gene cloning techniques based on E. coli as host. Many applications of genetic engineering require vectors for the expression of genes on diverse eukaryotic organisms for engineering of new plants, gene therapy and large-scale production of eukaryotic proteins. Animal and plant-based vectors have been designed for this purpose. 1. Plant viral vectors. Virus genome containing DNA insert cause viruses infection in host plant and amplified naturally. However, there are many viruses that causes systemic infection. Consequently, their genes are introduced into all the cells. Since virus infection results in delivery of a new genetic material into host cells, they provide a natural example of genetic engineer. The DNA insert linked with viral DNA that are present in host cell replicates and expresses in host cell along with viral genes. For insertion of a desired gene into viral genome, it is necessary to study and characterize the virus into detail so that the DNA linked with virus genome may express in the plant. Construction of viral vector that can express in host cell is based on genomic DNA or RNA molecules. Plant virus vectors are non-integrative and transmitted systematically in plant and assembled in host cell in high copy number. But it never integrates with the host genome. Therefore, to act as viral vector, they should 1. Carry extra nucleic acid, DNA or RNA, 2. Have broad host range, 3. Be easily transmitted, and 4. Be preferred to manipulate their genome. There are 3 groups of viruses: Tobamoviruses, Gemini Viruses and Tobamoviruses, which have been worked out in detail. See the figure. Figure 5.18. A genomic map of Cauliflower mosaic virus. The dark boxes are the coding region. DNA is in the center as thin line and outside as 35S transcript. A. Tobamoviruses. Cauliflower mosaic virus CaMV, commonly known as Tobamovirus, which has extensively been studied. This virus infects mainly the members of Cruciferae. Genome of CaMV is made up of double-stranded DNA which was first manipulated by recombinant DNA technology. The CaMV is transmitted naturally simply by rubbing of leaves or by aphids. Viral particles transmitted are isometric, 50 nanometers in diameter and isolated from inclusion bodies. Viral particles spread rapidly in 3 to 4 weeks throughout the plant and found at high copy number, about 106 virions per cell. Genome of the CaMV is a linear, open, circular molecule of 8 kilobits twisted or knotted forms, figure 5.18. It consists of tightly packed 6 major and 2 minor, total IVIII, open reading frame, on one coding strand. It replicates through reverse transcription. After infecting the host cell, DNA enters inside the nucleus where a supercoiled minichromosome is formed after digestion of a single-stranded overhangs and digestion of gaps. Inside the nucleus of plant cell, the minichromosome acts as a template for nuclear RNA polymerase 11 forming RNA transcripts. Then the transcripts are transported to cytoplasm where it undergoes either translation or replication into DNA strand using reverse transcriptase. The plus DNA strand is synthesized from two primer binding sites that are present near gaps 2 and 3. The RNA synthesis starts from gap 3' to 5' end of DNA strand, whereas synthesis started from gap 3 continues to gap 2. Finally, DNA molecules are packed to form viral particles. These particles restart the similar events after re-entering the cell of a healthy tissue of other plants. There are two non-essential genomic regions of the virus. ORF-I and ORF-V11 are non-essential. ORF-11 synthesizes the factors for insect transmission and ORF-7 has unknown function. These two regions can be replaced by a foreign DNA and vector constructed. By using this approach, in 1984, Brisson and co-workers transferred CaMV vector containing the bacterial dihydrofolate reductase into turnip plant cells. This gene is expressed into the plants. B. Geminiviruses. This group consists of single-stranded DNA containing plant viruses that cause diseases in several plants. It has geminated two paired particles, morphology which differentiate it from the other groups of viruses. The capsids are icosahedral, of 10 to 20 x 2 nanometer dimension enclosing covalently closed 1 or 2 circular single-stranded DNA molecules, 2.6, 3.0 kilobits. After infection, DNA molecule enters in the nucleus. The single-stranded DNA is converted into a double-stranded DNA replicative form. Thereafter, many copies of replicative forms of DNA is formed inside the nucleus, possibly through rolling circle mechanism. Gemini viruses consists of two elements, i.e., an origin of replication and replication protein. Each may become a part of vector. The geminivirus vectors which have been constructed so far consists of reporter or other desired genes, replacing the coat protein expressing sequence of viral genome. These vectors insert foreign gene in protoplasts, cultured cells or leaf discs of desired plants. 3. Tobamoviruses. Tobacco mosaic virus (TMV) consists of RNA as a linear, large, undivided genome. RNA-based vector is constructed by inserting foreign gene into an intact viral genome. For example, TB2 was the first RNA-based RNA vector which was successfully spread in whole plant, Fig. 5.19. TB2 vector was constructed by inserting a foreign gene at the 3' end of the movement protein ORF. The native subgenome promoter, SGP, drives the expression of the foreign gene under SGP from the related virus IORSV. Odontoglossum ring spot virus governs the expression of ORF of coat protein. RNA viral vectors containing gene insert can replicate only in protoplast, inoculated leaves or differentiated tissue. For detailed description see Advanced Biotechnology by RCW, 2014, see the figure. Fig. 51.19, Genetic map of tobacco mosaic virus-based TB2 plant RNA viral vector, insert equals NPT2, a trichoxanthin, viral coat protein, MP equals viral movement protein, representative equals replication. 2. Animal viral vectors. In nature there are several viruses which cause diseases. A virus gets adsorbed to the body surface of suitable host and infects the cell. This ability of animal viruses has been exploited and virus-based vectors have been designed to introduce foreign DNA of known function into cultured eukaryotic cell. In 1979, for the first time a simian virus 40, SV40, based cloning vector was constructed and used in cloning experiment using mammalian cells. Since then several vectors were constructed using adenovirus, papillomaviruses, retroviruses to clone foreign DNA into the mammalian cells, and baculoviruses in insect cells. In recent years, retrovirus-based vectors are commonly used for gene cloning, because they can infect several types of cells. Since they are single-stranded, their high concentrations are required for fully differentiated cells like neurons, hepatocytes, etc. The GAG, POL and ENV genes of retroviruses, which are required for replication and assembly of viral particles, can be replaced with foreign DNA. The recombinant DNA is introduced into mammalian cells in tissue culture, Figure 5.20. For detailed description see Advanced Biotechnology by RCW, 2014. See the figure. Figure 5.20. Construction of retroviral genome to be used as cloning vector. Problem statement. End of the chapter 5.